Good morning, students. Let's start and continue our biomolecules chapter. Biomolecules. Biomolecules, as we have already studied, that lipids, nucleic acids, and carbohydrates and proteins, these are the biomolecules. Out of which we have done lipids and nucleic acid in the last lecture. Now we will do carbohydrates and proteins. So our first topic is carbohydrate. Carbohydrates, that means polysaccharides. Carbohydrates are the acid insoluble substance they are called polysaccharides. Poly means many and saccharides means sugar units. Polysaccharides, they are generally have the monomer unit as saccharum. Carbohydrates, which have only single unit, they have monosaccharides. One monomer. Disaccharides having two monomers. And polysaccharides have many monomer units. So polysaccharides में हमारे पास आते हैं cellulose like starch. In monosaccharides, they are the generally we can see glucose, fructose. These are 6 carbon compound and ribose. This is a 5 carbon compound. So these are the monosaccharides. In this, 2 monomer units are there. Like examples are galactose. In polysaccharides, there are the many monomer units. More glucose are present, and the example is starch and cellulose. So polysaccharides examples are here: starch, glycogen, and cellulose. What is the difference between them? These beads are showing the monomer unit. These monomers are arranged in a single line in starch. In cellulose, these monomer units are arranged as a sheets interconnected. And in glycogen, where the branching is present in monomer units like glucose. So glucose that generally have formula this one. So in this way, 6 carbons are present and hydroxyl group is present in each of them. At each carbon, hydroxyl group is present. So this is the formula of glucose. In this, glucose and glucose, 2 units are connected with each other. By losing H2O. So after dehydration, glycosidic bond is formed and make a disaccharide. and they get connected with each other, minus H2O, and they are connected with a glycosidic bond. In this way. So this is the glycosidic bond present. This glycopolysaccharic bond can be seen in this form. Here, the glucose molecules are joined with the help of glycosidic bond by removing water. By removing water, the two glucose units and more glucose units, they are connected and form a branch structure that is called starch, which is a polysaccharide. So, in this way, polysaccharides are formed. Starch has helical structure, so can hold I2 molecule, while cellulose does not have helical structure. So it cannot hold I2, and because of that it gives blue color with iodine and it doesn't give blue color with the iodine. So this is the difference why starch gives blue color in the presence of iodine and cellulose doesn't give color blue in the presence of iodine. क्योंकि स्टार्च जो है वो हेलिकल स्ट्रक्चर में होगा और उसके अंदर जो I2 है मॉलिक्यूल वो आपका होल्ड हो सकता है, बट सेल्यूलोज में नहीं हो सकता। इसके अलावा और भी हमारे पास पॉलीसेक्राइड्स हैं, जिसके एग्जांपल्स हैं। नॉर्मली पॉलीसेक्राइड्स आर होमोपॉलीमर, having similar monomer unit. But some heteropolymers are also there. Example, chitin present in exoskeleton of cockroach. Another example is one amino sugar having amino group. Example, glucosamine. So, polysaccharides can be homopolymer and as well as heteropolymers also. If some other functional groups are present, like amino group in the glucosamine, and there's another example, N-acetyl galactosamine. So, this is the polysaccharide structure. Now, we will do the next polymer, that is protein. Protein, it is a heteropolymer. That is made up of different monomer units. And that monomer unit is called amino acid. There are 20 types of amino acids. 20 different types of amino acids are present, interconnected with each other with the help of peptide bond. A carbon atom having amino group on the one side and carboxyl group on the other side, and one having hydroxyl group, hydrogen, and another side there is the carbon atom present with a different number of carbons. So You can see here R representing the side chain. This one is R और यहाँ पे एक अमीनो ग्रुप है, एक तरफ कार्बोक्सिलिक ग्रुप है, one there is hydrogen, another side is साइड चेन. तो एक कार्बन से चारों तरफ से अलग अलग जो है हमारे पास जो मॉलिक्यूल जुड़े हुए हैं, तो इसलिए इस स्ट्रक्चर को हम ड्यूटेराइन स्ट्रक्चर बोलेंगे. ड्यूटेराइन स्ट्रक्चर में यहाँ पर अगर एक H+ हो जाए तो इसे हम पॉजिटिव चार्ज आयन कहेंगे. और यहाँ पे अगर इन द सेम वे अगर यहाँ पे जो एक हाइड्रोजन कम हो जाए तो यहाँ पे नेगेटिव चार्ज आ जाता है. तो इसको हम तो इसमें जो है पॉजिटिव और नेगेटिव दोनों चार्ज आ सकते हैं अमीनो एसिड पे. दिस इज फॉर नेगेटिव चार्ज, दिस इज पॉजिटिव चार्ज अमीनो एसिड. तो इसे हम ड्यूटेराइन स्ट्रक्चर कहते हैं बिकॉज इट कैन फॉर्म एनायन एंड कैटायन बोथ. नाउ proteins are formed by making peptide bond in which NH2 and other carbons carboxyl group they help in the formation of water molecule. and पानी का मोलिक्यूल जब एक रिमूव हो जाता है तो यहाँ पर हमारा पेप्टाइड बॉन्ड बनता है. So you can check it with the help of the diagram. You can see here the peptide bond is formed by removing hydroxyl group from one and hydrogen from the amino group of the two different amino acids. and पानी जो है वो माइनस हो जाएगा और यहाँ से हमारे पास पेप्टाइड बॉन्ड बन जाएगा जिसमें CO और NH रह जाता है. दिस इज कॉल्ड पेप्टाइड बॉन्ड. Now, in this way, proteins are formed. Now we can represent protein structure with the help of this diagram, in which primary structure show the sequence of amino acid in which they are joined. अलग अलग अमीनो एसिड कौन से सीक्वेंस में ज्वाइन है, 1 पहले लगा है, 3 बाद में लगा है, 2 पहले लगा है, तो उसके बेस पे इसको हम प्राइमरी स्ट्रक्चर बोलते हैं. सो इट इज जस्ट अ सिंगल चेन हैविंग डिफरेंट अमीनो एसिड. सो दिस वन इज शोइंग द प्राइमरी स्ट्रक्चर. नाउ सेकेंडरी स्ट्रक्चर. In secondary structure, these single chains are arranged in the form of helix. Or beta pleated sheet structures. So secondary structure is present as alpha helix and beta pleated sheet. You can see here this is the secondary structure जिसमें दो दिखाई है अल्फा हेलिक्स स्ट्रक्चर जो हेलिकल फॉर्म में है और शीट की तरह से अगर लेयर अरेंज हो जाती है तो उसको हम बीटा प्लीटेड शीट स्ट्रक्चर कहेंगे. नाउ देयर इज अ टर्शियरी स्ट्रक्चर in which these helix coiled on itself or the beta pleated sheet say the coiled on itself. This is called tertiary structure. Quaternary structure including a number of these tertiary structures.